Welcome to this third meeting in our meeting series about advanced therapeutics and medicinal products, or ATMP in short. This meeting series is a collaboration between the Norwegian Medicines Agency and the Norwegian Pharma Trade Organization. And my name is Hege Edvardsen, and I'm the Senior Manager in LMI for Research, Development and Innovation, as well as ATMP. Today's meeting is our third meeting in the series and is focused on gene therapy. The presentations today will primarily be held in English, as we have international guests presenting the first two topics. First, we will get an overview of gene therapy for people with rare genetic disorders before we get some insights into it from a regulatory perspective. The last presentation will provide us with some insights into how the ongoing development is perceived by the clinicians. Questions to the presenters can be asked in the chat, and we will try to answer them after each session. As you could probably see, we are making a recording of this meeting, and we would ask all of you following the meeting to turn off your camera and microphone unless you are a presenter. So our first speaker today is Dr. Martin Schulz. Dr. Schulz is Senior Medical Director for the Gene Therapy platform at the Rare Diseases Global Medical Affairs team at Pfizer. He will provide us with some insight with regards to the current status of gene therapy for people with rare genetic disorders. Dr. Schulz, welcome, and thank you for taking the time to present to us today.
Thank you very much, Hege, and to the organizers for inviting me. Very excited to be here at this early hour. I hope everyone has their coffee ready. Let me share my screen. Can you all see my slides?
Yes, we can see them perfectly. Thank you.
Perfect. All right. So as Hege said, my talk will be on gene therapy for people with rare genetic disorders. I will provide an overview talk. Of course, there is a lot because I only have half an hour that I will need to brush over a little bit. So I hope you forgive me for that. And just as a disclaimer, I'm an employee of Pfizer. Now, these are the key messages of my talk. So most rare disorders are genetic in origin, and gene therapy really aims to target the underlying genetic cause of that at a cell level. Now, gene therapy has the potential to offer long-term transformative benefits to patients. However, it's important to note that gene therapy is not an appropriate treatment option for every patient with a certain genetic disorder. And to realize the promise of gene therapy, really all stakeholders, and that involves patient groups and physicians and other healthcare professionals, as well as regulators, payers, and policymakers and the biopharma industry will need to work together to find sustainable solutions that enable patient access. Now, why is gene therapy particularly interesting for patients with rare genetic disorders? Well, we have an estimated 400 million people worldwide that are affected by a rare disease. So there are 7,000 known rare diseases, and only 5% of these have available treatment options. But 80% of these rare diseases are genetic in origin and could become potential targets for gene therapy in the future. Now, what's the concept of gene therapy in a nutshell? Well, gene therapy is a new generation of medicine where a functioning gene is delivered to the target tissue in order for the body to be able to produce a missing or non-functioning protein. So by using genes as medicine, we can target the underlying cause of a disease at a cell level, potentially with a single treatment. So gene therapy possesses the potential to offer long-term transformative benefits for patients with rare disorders, and it can potentially eliminate the need for ongoing therapies and reduce the burden of frequent disease management. Now, this is an overview of genetic medicine. And what you can see here is on the left side, in the left column, you see gene transfer. So this is in vivo gene therapy and also ex vivo gene therapy. So it's called in vivo gene transfer and ex vivo gene transfer. So let me start with in vivo gene therapy. So this is the use of a carrier to directly deliver a gene into the target cells. And this can, for example, be a recombinant AAV vector, and I will talk a bit more about that later on. And then there's also ex vivo gene therapy. This is where patient cells, often that's hematopoietic stem cells, they are isolated from the patient. Then they are transduced in the lab with a desired gene, and then they are reinfused back into the patient. Often lentiviral vectors are used there. And really the purpose, the objective of gene transfer is to add a functioning gene. Now in the middle column, we've got gene editing. And that is really to make precise changes of the patient's own DNA using site-specific targeted nucleases. So I think probably most people have heard about CRISPR-Cas9, maybe about zinc finger. And what that really relies on is these nucleases and then the cell's own repair mechanisms. And the objective of gene editing is to permanently remove, modify, or add a functional gene. And then on the very right side, we've got gene regulation. So that's the change in expression of a gene by targeting RNA. So we are targeting the product of DNA transcription with, for example, antisense oligonucleotides, small interfering or microRNA. And the goal here is really to silence or enhance gene expression. Now, for this talk, I will focus on in vivo gene therapy with recombinant AAV and a little bit on gene editing. I believe that ex vivo gene therapy will be covered in another talk in this webinar series. So in vivo recombinant AAV gene therapy is Pfizer's main approach to gene therapy. It's really the delivery of recombinant AAV vectors, and AAV stands for adeno-associated virus. So these are vectors that are based on adeno-associated virus to transfer a functioning copy of a gene into the cells of a patient to treat a specific genetic disease. So this is in vivo recombinant AAV gene therapy in a nutshell. So what it really depends on is effective vehicles that transport the functioning gene into the target cells. What we have, again, are vectors that are based on and modeled after AAV, after adeno-associated virus, but they do not contain the viral genes anymore that code for proteins. They really contain the therapeutic DNA, the transgene, the functioning gene. And this is then this vector is then infused into the patient. So this can either be done systemically through the bloodstream, and then the vectors hone into the target cells, or this can be done locally, for example, into the eye. The cells that we transduce, the target cells, they should be long-lived post-mitotic cells to enable long-term gene expression, because the transgene after transduction will exist mostly in an episome in the nucleus. So for the most part, it will not integrate into the host genome. So when the cells divide, there's a good chance that it would be lost. And this functioning gene is then used as a blueprint to produce the missing or non-functioning protein. And what you can see here is that there are different AAV serotypes that have a different tissue specificity. So we can use them to do a certain degree of tissue targeting, depending on the disorder we would like to treat. Now, again, the success of gene therapy really depends on effective vehicles for gene transfer. So we've got the capsid, so that's the protein shell that's modeled after a virus, in this case, AAV, that protects the genetic material while it's in the body. And that helps it to hone into the target cell, as we've seen. Then we've got the transgene in the transgene cassette. So that's really the DNA, the functioning DNA that will be introduced into the target cells. Both of them form a vector. So a vector is the transgene encapsulated into a capsid. And these vectors, again, are based on viral platforms, but they are not viruses. They are then infused into the body, where they hone into the target cells. So, this could be, for example, the hepatocytes. So how does recombinant AAV vector deliver the transgene to the target cells? Well, it's most of the time, as you would have expected, it starts with receptor binding. So it's certain proteoglycans on the surface of the target cells where the vector binds and attaches. Then the next step is endocytosis. So the vector is taken up into the target cell by endocytosis. Then it develops, the endosome develops from an early to a late endosome. The pH changes, and the vector is then delivered to the cell nucleus. In the cell nucleus, uncoating is happening. So the capsid is removed and broken down, and the genetic material is released. The genetic material in the beginning is single-stranded DNA, so that is then converted into double-stranded DNA by the cell, and then it's there to be transcribed into mRNA, and then later on translated into the protein that has been missing. And as I said earlier, the transgene is mostly maintained as a ring-form structure, as an episome in the nucleus of the cell. Now, what makes AAV particularly amenable as a platform for the production of recombinant vectors to be used in gene therapy? Well, it's a number of things. So the first one is a lack of pathogenicity. So AAV is not known to cause any disease in humans. Second one is the potential to establish a long-term transgene expression. So again, we are targeting long-lived cells, so muscle cells, hepatocytes. We're targeting cells that are post-mitotic, and there the hope is that we can see long-term transgene expression. There's defective replication. As I said, we are taking out the viral gene, so that the vector cannot replicate. And also AAV needs helper viruses in order to replicate. So there's defective replication. As I said earlier, there are multi-serotypes of AAV that permit a broad tissue specificity. So there's the possibility of a certain tissue targeting. These vectors are predominantly non-integrating, so they predominantly do not integrate into the host genome. They exist as this ring-like episomal structure. And also compared with other vector platforms, AAV is relatively non-inflammatory. Now, it's important to note that whilst this is very exciting, AAV is not all patients will be eligible for recombinant AAV therapy. And here are some factors that impact the eligibility for recombinant AAV gene therapy. So when you start with the total population with a certain genetic disorder, then some of these will have had exposure to wild-type AAV in the past. So I've talked about that. This is clinically silent, but they will have developed antibodies against AAV, and then they might not be eligible for the therapy anymore. Disease progression plays a role. Once the disease has progressed too far, patients might also not be eligible for gene therapy anymore. The patient's age plays a role, and depending on the therapy, sometimes there's a certain age that the patient needs to reach to receive the therapy, and sometimes it's the other way around. Comorbidities play a role. So, for example, severe liver disease, active hepatitis B and C, these are common exclusion criteria in the trials that we see with recombinant AAV gene therapy. So in the end, we end up with an eligible patient population that is quite a bit smaller than the total population with a genetic disease that we've started with. Now, here are some immunologic considerations for recombinant AAV gene therapy, and they impact both the effective transduction of cells, and they also play a role when we want to establish really a transduced cell population, a stable cell population. So let me start with a humoral immunity, and what humoral immunity really means these are antibodies. So preformed neutralizing antibodies to the recombinant AAV vector can exist in a patient as a result of a clinically silent infection with AAV, so the patient doesn't know that. But these antibodies can prevent transduction by binding to the capsid before it can transduce the cells, so they can really prevent the therapy from being effective. And humoral immunity, these antibodies can also prevent retreatment, because once you've received treatment with a recombinant AAV vector, you will mount a pretty strong antibody response against this recombinant AAV vector. I should say there's a lot of research going on in order to mitigate these effects. So there's, for example, research going on in the field of plasmapheresis to try to get rid of these antibodies before you administer the therapy to the patient. There are also enzymes that can break down IgG antibodies before the therapy. So that one of the enzymes is called IDAS. So there's a lot of research ongoing in order to try to mitigate that. On the other hand, we've got the cellular immunity. And what the cellular immunity, as the name says, it's really a cytotoxic T cell response. So you can see here, this is what happens in the cell. The vector is taken up by the cell, and then the capsid is broken down into epitopes. And these epitopes of the capsid, they are displayed by so-called MHC class 1 at the surface of the cell, and then T cells can respond to these capsid epitopes that they see, and then they would attack and kill the transduced cells, because basically what they think is that this cell has been infected by a virus. And of course, when that happens, that means that the very cells that you were able to transduce with your gene therapy, that they will be eliminated. And of course, that's something to be avoided, because that can result in a potential loss of transgene expression. So again, there are good strategies to mitigate that. A lot of the clinical trial protocols have immunosuppression with corticosteroids and others in order to mitigate for this effect and tone down the T cell response. Now, here's some further considerations with regard to recombinant AAV gene therapy, I think, that are particularly important when having conversations with patients about gene therapy. So the first one is that the results may be variable. So the effect might not be the same in every patient, and there may be some patients where the effect that is seen is so that they still have symptoms, or they still need treatment. There is long-term follow-up required. That is very important in order to monitor for long-term durability, but also of course, for safety, and in particular for safety events that have a very low incidence. So when you think about potential cancer development. So that is really important. It's a big commitment for the patient. Long-term follow-up is required. And ideally, I think this should be lifelong follow-up after receiving the therapy. Some gene therapies are limited to a one-shot treatment. So I've talked about that earlier. This is because patients will mount an antibody response after gene therapy. The genetic disorder can still be passed on to the children, so we are not changing the germline. The long-term durability of clinical benefit is yet to be established. There is some encouraging data, but the really long-term and potentially lifelong durability of some gene therapies still needs to be established by data. And then there is a certain vector shedding potential. So in the bodily fluids, the patient after gene therapy will shed the vector. There is the theoretical concern that this could be transmitted then to, for example, family members or the environment. So it's important to make the patient aware of that. And also after gene therapy, often there is a need for barrier contraception after gene therapy, just as a safety precaution. Now I switch gears a little bit. So this was recombinant AAV gene therapy, and now I'll talk a little bit about gene editing. And gene editing can be defined as modifying part of a gene using gene editing technology to remove repeated or faulty elements of a gene, or to replace a damaged or dysfunctional region of DNA. And I think what that really means is that gene editing, in other words, can be used to do really a variety of changes to the host DNA, and that starts from inactivating a gene to introducing a new gene, and even, as we'll see, there's the possibility with base and prime editing to do small changes of individual bases or base sequences. I will say that in contrast to recombinant AAV gene therapy, the field of gene editing is quite new. I think with recombinant AAV gene therapy, of course, we've got some marketed products and we've got experience in clinical trials of over 20 years. And in contrast to that, gene editing is quite a new field. Now, what are the core principles of gene editing? Well, all genome editing nucleases, so the CRISPR-Cas9 system, TALEN, zinc finger nucleases, they all have a sequence-specific binding domain and they have a cleaving domain. So the binding domain really looks for the right part in the host DNA where the cleaving, the cut, you see the scissors here, should take place. So this is the first part. The second part is that gene editing relies on the cell's DNA double-strand break repair mechanisms. So we've got the endonucleases. What the genome editing endonucleases do is really they make the cut, you know, at the right side, and then the cell's own repair mechanisms, they do the rest. And of course, the aim is to allow for a specific beneficial change at the intended genomic site only. However, I think it should be mentioned that all the nucleases that we have, they are not perfectionists. So there will always be a certain degree of off-target editing. So gene editing really relies on the DNA double-strand break repair mechanisms of the cell. So again, here we've got the DNA, and then we've got the gene editing nucleases that create a double-strand break. And then there are really two pathways that the cell has. There are two repair mechanisms that the cell has in order to repair the DNA. And one is the really the non-homologous end joining pathway, the NHEJ pathway. And what that does is it fixes the DNA, but there will be some small changes to the DNA. So there will be some small insertions or deletions, what is called indels, and that will result in gene disruption, in gene knockout, in the inactivation of this gene. So that's the one pathway. And then there's the other pathway, which is the homology-directed repair pathway. And this pathway can happen when there's a template DNA that is provided to the cell that where basically at the cut site, the end sequence of this template will fit into the cut site. And then what can happen is that this gene is inserted into the DNA. So these are two different pathways. And when you think about disorders, then for some disorders, you might want a gene, really a gene inactivation. For example, when you think about Huntington's disease, and then for others, of course, you want the insertion of a functioning gene. Now we'll talk a little bit more about the CRISPR-Cas9 system, just because, you know, everyone is, of course, talking about it. Everyone's aware of it after the 2020 Nobel Prize of Jennifer Doudna and Emmanuel Charpentier, and how that works. And what you see here on the right-hand side is that what this system has. It has a guide RNA, a gRNA that tries to match the host DNA. So this is a sequence of 20 base pairs, and where this guide RNA pairs with the host DNA, where these 20 nucleotides pair, this is where the Cas9 endonuclease then cuts the DNA. Now, the CRISPR-Cas9 system does not search everywhere in the host DNA. It searches at certain regions that are close to a palm sequence in the host DNA. So that stands for protospacer adjacent motif, and that is for the CRISPR-Cas9 system. That is a base triplet, where the second and the third triplet is a guanine. Now, what really makes CRISPR-Cas9 quite different from zinc finger and TALEN is that for CRISPR-Cas9, RNA is used as a recognition molecule. So you can see here, it's really this guide RNA that is the recognition molecule. And of course, it's relatively easy to produce a guide RNA. for zinc finger and talon, you've got protein that is the recognition molecule, and that really requires a lot more. That is a lot harder to manufacture and a lot harder to change. And here with CRISPR-Cas9, for a different site you would like to cut, you can very easily just change the guide RNA. Now, I will not have time to go into recent developments. The whole gene editing and field is a hotspot for research. So apart from what I've shown here, there are base editors that can edit single bases in the host DNA, and prime editors that can make a little bit larger changes, and both of them can do that without double-strand breaks, but with just nicking one of the strands of the DNA. And then there are epigenetic editing approaches that are also very exciting, but I think they are a little bit out of scope of today's presentation. Now, I just want to touch on the current main challenges in the field of gene editing, and one is increasing the specificity of gene editing. So we really want to limit the off-target effects of gene editing, obviously for safety reasons. And then in the middle, we've got improving the efficiency of gene editing. So of course, when it comes to on-targeting gene editing, we want to see very high levels of on-target editing. And then I just touch on the delivery system. So gene editing is often used at the moment in an ex vivo fashion, so in cells that have been taken outside of the body. But in particular, when we think about going in vivo with gene editing, the delivery system is really a field where where challenges exist, just because you need to provide the nucleases or the mRNA of the nucleases. You need to provide the guide RNA, and potentially you need to provide a DNA template as well. And you need to think about something that has the packaging capacity to really deliver that to the cells, and also the tropism for it to arrive at the right cells. So there's a lot of work ongoing, and sometimes there are different delivery systems are used. So for example, a combination of recombinant AAV and lipid nanoparticles, but there's still a lot of work that needs to be done in that space. And with that, I come to the last part of my presentation, which is really on patient access to gene therapy. And the first thing I would like to say is that the engagement with the patient community is absolutely critical for drug development in rare diseases and in gene therapy. In particular, I would like to touch on that this engagement with the patient community, who really know their disease best, is important, from early research optimization to clinical trials, to regulatory and payer engagement, disease education, care considerations, and policy advancements, to really make sure that therapies are developed that the patients need, want, have access to, and will adhere to. Now, gene therapy may potentially offer greater one-time clinical benefits than those offered by conventional therapies, and this might mean for their patients and caregivers, this represents the potential to avoid years and potentially a lifetime of burden associated with chronic disease management and living with a severely debilitating or even life-threatening disease. And for payers, this means that potential long-lasting benefits could reduce or even eliminate the ongoing cost of disease management. Now, the recent scientific and medical advances in the field of gene therapy, they've been substantial. However, our healthcare system needs to evolve, so the science is not outpacing the policy development, and that needs to happen in order to really bring these potential gene therapy breakthroughs to the patients. And I would just touch on 4 areas I think that need to evolve. So the first one is delivery to patients and long-term follow-up. So how can we ensure the best delivery of gene therapy to the patients and the required long-term follow-up there's a need for registries that might need to be identified or even built, and that is work that needs to start now to really pave the way for gene therapies in the future. The second point is on value assessment. So how do value assessment frameworks for therapies need to be evolved in order to account for potentially transformative one-time treatments with a long-term effect? And part of this discussion are also value-based agreements that connect the reimbursement to how well a certain gene therapy is doing in a patient over time. The third part is regulations. There are certain regulations that might need to be adjusted to account for gene therapy. So, for example, the regulations for genetically modified organisms, GMO. And then last, but of course not least, we need centers to be ready to provide gene therapy and the required follow-up after administration and after approval of these gene therapies. So in order for them to be ready, they of course need the right procedures in place, the right education, and also the right facilities. Now we'll close with my key messages that most rare disorders are genetic in origin, and gene therapy aims to target the underlying genetic cause at a cell level. Gene therapy has the potential to offer long-term transformative benefits to patients. However, gene therapy is certainly not an appropriate treatment option for every patient with a certain genetic disorder. And to realize the promise of gene therapy, really all stakeholders, and I think a lot of them we have in the room today, will need to work together to find sustainable solutions that enable patient access. And I will close with my thank you slide, and really on the left hand side there the rare disease patients that inspire our work every day. And I would like to thank you for your attention, and yeah, I'd be very happy to take a few questions.
Thank you for your very, very clear and thorough presentation of gene therapy. I have not seen any questions in the chat as of yet, but please just put them forward in the chat if you have questions to Dr. Schultz. I would like to ask you one question though, Martin. So as of today, there's only a couple of handfuls of products available that have been going through the approval system and been approved in Europe today. But if you look at the pipeline, there's a lot of activities going on. So when do you, time-wise, when do you see this going from only a handful to being really a broad toolbox of gene therapies available for the rare disease community?
Yeah, thanks, Heike. That is, of course, a really good question that is very hard for anyone to answer without a crystal ball. And I will not be able to give you a number. But what I can say is that we've got really a substantial number of gene therapy investigational products in late stage. So I think over the next year, certainly this will pick up speed, and we'll see more approvals. We'll see more approvals in that field, and that is really why, as I tried to say in the last part of my presentation, why we need to ensure that we've got the right policies in place when they come, because we all know that policy changes take time. And so we really need to start now to work on these to be ready for these gene therapies in the future.
I think that's a very good point. And I also like your point about working together to ensure that we have registries available that will be able to follow this, to be used for following up the patients over time. I think that's also a very central aspect. So you will stay on the line for a bit if there are questions pop up. Perfect. So our second speaker today is Dr. Carla Herberts. She has extensive regulatory experience working at the Dutch Medicines Agency, and where she has mainly been focusing on immunological and cell therapy products, as well as hemato-oncology products. She is a Dutch representative in the EMA's Committee for Advanced Therapeutics, or CAT, and she has acted as the EMA rapporteur in the approval of a number of the new ATMPs. So Dr. Herberts will give us the regulatory perspective on gene therapy. Welcome, and we were very much looking forward to hearing your presentation.
Thank you for the introduction, and I have to apologize. I will not be on camera somehow. I cannot get my camera on working in this team meeting.
We hear you perfectly.
That's good. That's good to know.
Do you want me to share the slides, or will you manage to share?
I hope I can manage. If there's in the presentation mode, yes.
You don't see your slides yet, Carla.
You do see them, right?
No.
Not yet.
No.
I can see them. Then maybe you should share them, then try to avoid all the hiccups.
No, that was the Pfizer one. So let's share that one. Let's share the one for you.
Thank you.
That we had already heard. So let's see.
Yes, I would like to thank Martin for this excellent presentation and the starting presentation for this meeting. I think I can omit some things I wanted to say on the AAVs before, because he very nicely explained how AAVs work, because this is one of the examples I wanted to give.
So now I think your slide should be on the screen.
Yes, thank you. Thank you. So as mentioned, I am from the regulatory field. And as a regulatory person from the regulatory field, I always have to make this disclaimer that the views expressed are my personal views and not necessarily reflecting anything my employer, CAT or EMA. Thank you. Next slide, please. So from a regulatory perspective, it's very important what a gene therapy medicinal product actually is. So I listed here the definition, which is the current definition of gene therapy. We are brainstorming on a new legislation and also on a new definition for gene therapy, but for now it is like this, that it should contain a recombinant nucleic acid administered with a view to regulate, replace, repair, adding or deleting a genetic sequence. And the therapeutic, prophylactic or diagnostic effect directly relate to the recombinant nucleic acid. So next slide, please. So the main, let's say, two divisions on gene therapy are in vivo gene therapy and ex vivo gene therapy. Those are the type of gene therapies we see. And this can be either done with a viral delivery system, as for example, AAVs or the lentiviral vectors, which are used in ex vivo gene therapy, but also other delivery systems and gene editing can also result in gene therapy medicinal products. Next slide, please. So from a regulatory point of view, what are gene therapies? Well, they are classified as an advanced therapy medicinal products about 10 years ago. And as you all know, all advanced therapy medicinal products are medicinal products. This means that they require a marketing authorization. And for this, you need to demonstrate the quality, safety, and efficacy of a product and comply with GMP and GCP regulations. And there's a need for pharmacovigilance and risk management, risk minimization measures and risk management plan. All 80, for me, the slides have this bit. Yes, there it is again. All ATMPs are biologicals, and all ATMPs has to be centralized, has an application for a marketing authorization has go through the centralized procedure. And so you apply for a marketing authorization for whole Europe all at once. And the evaluation is done by the Committee for Advanced Therapies, and of course, CGMP is also involved. But this is for all products, and for ATMPs, it's specifically assessed by the Committee for Advanced Therapies. Next slide, please. So what makes ATMP different from other products? This is that their initiation and often also the first stage one or couple of stages of the clinical development is done in academia. So there's a relatively long academic development before the product comes to us, let's say. All the manufacturing of ATMPs are quite complex when you compare them to other type of medicinal products. They're difficult to characterize, and as a consequence, it's also quite complex to assess the impact of manufacturing processes or changes in the manufacturing process on the product. The batches are often very small, up to 1 batch per patient. So these autologous products have 1 batch per patient. And this is quite different from, let's say, an aspirin, where there's like 10,000 or more patients per batch. Because of the small batches and also sometimes the autologous nature, there's a higher variability in the batches and possibly also in the effect of the product. For some products, we need bedside preparation. Of course, this is not completely new, but if you think about cell therapy, medicinal products, or genetically modified cells, which are gene therapies, also there, the bedside preparation is very critical so that you do not lose the cells. As they are different from other products, or kind of different from other products, there is a specific GMP for ATMPs, in which there is a bit more flexibility with regards to the risk management, let's say, or the risk assessment of these products. And for example, out of specs batches can be given to a patient after a thorough risk assessment, which is not allowed for other type of products. If you look to the clinic, then ATMPs are often developed for small patient population. That's already said at the beginning, and this is actually also, I think, why we are here for the very small patient population. Or this meeting is for small patient populations, the orphan disease, monogenetic diseases, and these are not big patient populations. So this requires sometimes a different clinical approach. We often expect a large treatment effect. This has been observed, but this is not true for all ATMPs. Also, the duration of effect, it's often considered as a one-time treatment, which must have a very long or sufficiently long duration of effect, hopefully lifelong. So this is different from other products which you can administer multiple times, and the duration of effect is related to the duration of the moments you give the product to the patient. Next slide, please. So to make it a bit more interesting, I would say, I thought I would give you an example of the first AAV registered, one of the first AAV registered gene therapy medicinal products, which is Zolgensma, and I'm the rapporteur for this one. So Zolgensma is a gene addition therapy designed to introduce the wild-type copy of the defective prime gene, the SMN gene, for patients with spinal muscular atrophy. I have on the next slide, I have something more on the disease. It's an AAV 9 viral capsid, so this is the vector, and it's indicated for patients with spinal muscular atrophy with a biallelic mutation in the SMN1 gene and a clinical diagnosis of SMA type 1, or patients with a biallelic mutation in the SMN1 gene and up to 3 copies of the SMN2 gene. I will tell you a bit more next slide. And the dose is determined by the patient's body weight, and it's quite high. It's 10 to the 14th viral particles of genome copies per kilogram. So you can imagine that there's quite a high, let's say, viremia for these patients at the moment of administration. But as already indicated before, as AAVs are considered relatively safe, you can give such high doses to patients. We will see in the safety specs later. Next slide, please. So what is the disease? Because that's the thing this product is for. And patients with spinal muscular atrophy have a very dismal disease. This is the SMN genes are important for motor neuron survival. And if you have a biallelic mutation in the SMN1 gene, this can be counterfeited by the SMN2 gene. If you have, the more copies you have of the SMN2 gene, there's a kind of a rescue of these neurons. And if the less copies of the SMN2 gene you have, the more severe disease you have. And if you have 1 or 2 copies of the SMN 2 gene and not more. Then you have SMA type 1, and these patients are show already a very severe disease after birth. So the diagnosis is usually made before the 6 months of age. These patients never gain the ability to sit, and they generally do not survive past 2 years of age. At least not when you and the survival might be prolonged if you have mechanical, ventilatory, and nutritional support. The diagnosis can be made clinically, but it can also be made based on genetic testing. But then you have to test the patient. You can test the patient after clinical symptoms occur. But I think now usually this starts at the neonatal screening, that you can do a genetic testing, and then you can already diagnose SMA. This also spinal muscular atrophy based on the deletion of the the SMN1 gene and the number of copies of the SMN2 gene. So it's a very severe disease. That's the main message here. And you can identify it based on symptoms or pre-symptomatically based on genetic testing. Next slide, please. So what did we get from for the marketing authorization? We received quite a lot of studies with the product in type 1 SMA patients. But if you can see that the number of patients in these studies are very limited. So the first clinical study, the dose escalation safety and efficacy study, had only 12 patients in it. And it is formally a dose escalation study. However, they ended up with the first dose. And this is done with process A product. And patients from this study roll over into a long-term follow-up study, LT001. Upon the first results of the 101 study were quite promising, but they had to do some product development still. So we had a product, process B product, which was studied in the subsequent study. And in this study, 22 patients were enrolled, which were diagnosed with SMN type 1. And this study was done in the US. Later on, the 302 study was opened in Europe. So this is the European counterpart of 303 study. And at the time of application, the 304 study was opened, and this were patients which were diagnosed pre-symptomatically based on their genetic screen. As I mentioned, process A, the first study was with process A. Subsequent development resulted in the process B product, and comparability between these two processes was not completely established. So we could not say that process A was the same as process B, and this is why the data from study 101 were considered only supportive. And the data from study 303, which was then the most which was then completed, all patients were followed up for the primary endpoint, was the pivotal study. And the 302 and the 304 studies were, let's say, supportive because they were quite in early stages and still ongoing, still recruiting even. In the next couple of slides, I will show you the data of the 303 study. There were two other studies. So the 102 study, which is a study with another route of administration. So we did not assess this one. And there was the long-term follow-up study 002 study, which was in which patients of let's say the 300 studies would roll over once they have finished that study. These were all single-arm studies, so to contextualize the data, the company also submitted natural history studies from two different registries, the PNCR and the Neuronext registries, which had, I haven't listed it here, I think 23 and 19 patients each in matching the patients in the clinical trials. Next slide, please. So what was the design of the 303 study, which is an open-label, single-arm, single-dose study. There were 22 patients with type 1 SMA in the study. They could be symptomatic or pre-symptomatic. I must say most were symptomatic, and they could only have 1 or 2 copies of the SMN2 gene, and they had to be not older than 6 months at the time of infusion. So patients should not be too far in their disease process when they got the product. The primary endpoint, so what we were looking for in these patients was the number of patients which achieved functional independent sitting and survival at 40 months of age without substantial ventilatory support. And there were quite a number of other endpoints more directed at the motor development, the fine and the large motor, neuromotor development. Next slide, please. So what were the data? So the primary analysis, survival at 40 months of age. From the 22 patients in the study, 20 patients survived this landmark. And if you look at the natural history cohort, and this is the PNCR database, from the 23 patients, only 5 achieved this endpoint. So 80% already passed away before 14 months of age. And achieving the milestone of sitting independently, there you see that 60% of the treated patient population achieved this milestone, while none of the patients of the natural history cohort achieved this milestone. And if you see in the other slide, you can see the number of milestones documented these patients achieved, and at what time post the treated patients achieved this. And this is quite different what we have seen in the natural history cohort. So what we can see here is that the primary endpoint has been met. I'm not sure what and also that there's a huge difference, a large treatment effect compared to the natural history cohorts. So clear efficacy for these patients. Next slide, please. So the company, so this is the pooled analysis of all patients with SMA type 1 with only 2 copies of SMN2. And in blue, so this is from all the studies they have assessed, except the 101 study, which has process A. And there you can see that the survival of these patients is very high when you compare these to the 2 different natural history cohorts. Again, confirming the effect size and the high efficacy of this product in these patients. Next slide, please. So what about the safety? In general, as already somehow could be expected based from what we know from AAVs, is that the safety profile in these treated patients, and we had up to 90 patients treated at this time at the assessment. The safety profile was mild and manageable. We saw transient increases in transaminases and thrombocytopenia, which is a known effect, what we often see in patients treated with AAVs. This is because the AAVs generally target or distribute to the liver, and the liver has to respond to this. And we also saw the effect of the immune response. So there was a pyrexia and effects on the hematological parameters. And we saw an effect of prednisolone. So these patients were pre-treated and also treated for the initial part after exposure to after the AAV has been given with prednisolone to reduce the immune response, also to counterfeit the effect on the liver and the transaminases, because if there's a huge immune response, then you could have severe liver damage. So this is why prednisolone was given upfront as prophylaxis. There were some uncertainties in the safety profile because we had a lack of control. So we also saw some other effects like GI effects, which in general were attributed to the underlying disease. As you can imagine, these patients have a different diagnosis of SMA type 1. They have a problem with feeding with their muscles. So some of the feeding ends up in the lung, and then you have, for example, pneumonia. These things we saw a lot of GI effects, which were generally attributed to the underlying condition, but of course, because we did not have a control, it is quite well, there is an uncertainty that some of these effects could be attributed to the or were caused by the product. Also, we had some concerns based on findings in animal studies. In animal studies, you see toxicity on the dorsal root ganglion, which is the ganglion in which the motor neurons enter let's say the central nervous system. We saw inflammation there, and we did not know what would happen in the clinic. We did not saw any signs of dorsal root ganglion toxicity, but as a lack of a control and patients with already motor neuron loss or more motor neural problems, it's quite difficult to exclude that there is something going on. So this is something which we are following actively. And also in the animal studies, we saw some cardiovascular toxicity, which we again did not see in the clinical studies, but we could not exclude any real effect. And of course, the duration of treatment, the long-term effects, there were still uncertainties. The patients in the 101 study were observed for the longest time, so we had some idea, but as this data was only supportive, there is still uncertainty, and also on the durability of the treatment effect. This still was a question because the patients were followed up for approximately 2 years, and they survived, but we need longer term follow-up as well. So this was kind of the uncertainties also for the safety and for the long-term effects. Next slide, please. So overall, based on this brief overview I presented, we have some more data, but still we considered the benefit risk positive. But we had to discuss what was the target population in relation to the knowledge we had at the time of marketing authorization. And we felt that we needed some more data. So they also, the company, the authorization was a conditional marketing authorization because we wanted more data to confirm the benefit risk. And in particular, patients with who were treated pre-symptomatically, data in these patients were very limited, both in number of patients, but also in the duration of follow-up. So we had some specific obligations we asked the company. Next slide, please. So regarding the indication, we had several issues for discussion. So should we only limit, we had most data in symptomatic patients and only very limited data in non-symptomatic patients, so patients who were treated, who were identified based on their genotype. However, so we discussed whether non-symptomatic patients should already be approved and in the indication. And in the end, as we saw some effects already in these patients, and based on the biological of the disease, so that you would expect that the results in the non-symptomatic patients should at least be similar, or may even be bigger in the non-symptomatic patients, because there has not been or the damage to the nervous system is more limited than in the symptomatic patients. So you do not need to restore the patients; you do need to maintain these nervous systems. So we decided that non-symptomatic patients could be included. We also discussed which genotype. Most patients had SMA type 1, but how many SMN2 copies are allowed? When are patients with how many SMN2 copies are needed for, or actually should be restricted to how many SMN2 copies to have the SMN type 1 disease, and also should we include SMN type, the number of copies for the symptomatic patients or not. We discussed age restriction. We had, of course, patients were treated, always treated before 6 months of age, and so there were some more, but I think these more were the most important one. And as you can see in the final indication, which I wrote down again here, is that we made 2 bullets. So those patients who had a clinical diagnosis of SMN-A type 1, which means that the disease has to occur clinically before the 6 months of age, irrespective of the type of the number of SMN2 copies, could be treated with the product. And for patients who were not yet symptomatic, they should have been diagnosed genetically with a biallelic mutation in the SMN1 gene, because this is a hallmark of SMA, and they could have up to 3 copies of SMA type, the SMN2 gene. And next slide, please. So, and what were the specific obligations? So we already had the final study report of the supportive study with 101, but we also wanted the study report of the final study report of the pivotal study, the final study report of the European counterpart, and also the final study report of the 304, which is the study in the pre-symptomatic children in which patients were treated before 6 weeks of age, so before clinical onset or expected clinical onset. So these final study reports we wanted to see before we could consider the data comprehensive, and the application or the marketing authorization could be considered a full marketing authorization. And then as a consequence of this conditional marketing authorization, the company has to apply for an annual renewal of the marketing authorization until the specific obligations are fulfilled. And in this annual renewal, we want the update of the studies, but also the update of the clinical experience in let's say in the clinical practice with these patients. And in the patients, a lot of patients entered in a registry, so are quite well followed up. Next slide, please. I'm always to my end, I think. So the open questions still for this product is the dorsal root ganglion findings. This is still a concern, and we are following it up, in particular with regard to safety. As what I have not discussed, but has been mentioned in the beginning, if you listen very carefully, AAVs generally do not integrate into the genome, but in a very inefficient way, they do integrate sometimes. And as we give such a high doses to these patients, these inserts, there might be a clinical relevant, well, let's say, no, not clinically relevant, but we are for sure expecting insertions into the human genome because of this high dose. Insertions in the human genome have an inherent risk or concern of changing the genome makeup, changing gene expression levels, and in particular transforming cells into potential oncogenic or tumorigenic cells. So we have not seen any of this, but it is based on theoretical grounds. If a vector inserts into the genome, if it happens in let's say an unlucky locus, this could transform a cell into a tumorigenic cell. Of course, the insertions could also occur in principle, in theory, in germline cells. This is expected to be even less efficient than in normal somatic cells, but still, based on theory, we cannot exclude this risk. And these are theoretical risks thus far, but these keep us quite busy in discussing how relevant these risks, clinically relevant these risks are, and how we should handle these. Of course, the target population is still not that much an open question now for Zolgensma. At least we have an indication. If the company applies for an extension of the indication, then it's they are very welcome. But for example, in the US, the target population has an age restriction, and these questions come up to us several times. But I think this is also more a general point for gene therapies. What is the target population, and when should you treat patients? At what age should they be only symptomatic patients, or should you treat them pre-symptomatically, for example? The best route of administration is still an issue for debate for this product, but also for other gene therapy products. There is a study ongoing with this product for intrathecal administration, so that you the nerve cells are targeted, let's say, better, and you might give a lower dose. But this is still an ongoing study, so this is an issue for discussion later on. And maintenance of efficacy, so we do not know how long the efficacy will maintain, as the product remains mainly episomal. During cell division, there may be a possible loss of the vector. Of course, these patients, these are most post-mitotic cells, but still, this is something we want to ensure, that there is the maintenance of efficacy, and the possibility for retreatment is also discussed and as already highlighted by the previous speaker, so I will not go into this. And also the pre-existing immunity, can we treat, or how to treat patients who are seropositive for the capsid factor is an issue for discussion for AAV factors. And there are some more, but I think these are the main ones. And I think I will go next slide. It brings me to the end of my presentation, and I would like to thank you for your attention. And if there are any questions, I'm happy to answer them if I can. Thank you.
Thank you, Dr. Herbert, for a very, very nice presentation and overview. We have a very quiet audience today. I think they find everything both of you have said very clear and concise, but I have a question as usual. So this area is the area where you have a high degree of smaller environments developing new therapies. So you have smaller companies, not only the larger companies. And I know from dialogue with some of the environments developing ATMPs in Norway, that they've found the regulatory process very extensive and a bit complicated. So can you? But I know that you have some support processes that are available when you come to a certain point. So could you highlight or discuss those briefly?
Yes, I did not include these in the slides. Of course, there is quite a number of ways we can support the developers. If you have not to find, for example, if you are not really finalized your lead finding on a very general level, you can go to the EMA for an ITF meeting. You can come if you really have a product and you have questions on the quality, or what type of non-clinical studies you need, or translate these into clinical trials and the design of the clinical trials. You can ask questions in scientific advice, and the scientific advice can be done at the European level, so at the European medicines agencies, but also generally all the national competent authorities also have national procedures, which might be less formal and more, at least in my experience, and allow for a good interaction. If the product is very promising, a company can apply for a prime designation, in which a rapporteur is already early designated to the product, which also facilitates the interaction and acquaintances between the rapporteur on the dossier and between the rapporteur and the developer. As these are for small patient populations, if you apply for an orphan designation, this will have another interaction at the European level, but also will reduce the fees for these interactions with regulatory authorities. And in our national institute, in addition for the formal national advice, we have also a specific advice for academia, which is, well, I cannot translate it right now, but this is a specific advice for academia and also for very small companies. They can approach us and have a more informal interactions, and we even have a pre-discussion which is a kind of a feet on the table, discuss your product with us, and then we can recommend what the appropriate routes are for regulatory interaction. And I know our inspectorate is also open for discussions, of, for example, in relation to GMP and how best to develop your, let's say, design your manufacturing space, let's say. So there are, I would say, please reach out, and we try to accommodate as much as possible. I know EMA is also very aware that much of these products are developed by academia or by small companies, and in particular also academia, they are getting, EMA is getting more open in supporting also academia in their development. So this is, thank you for asking this, because it's like a reach out to the companies and the developers. Please contact us, because we want to support you as much as possible in the context that we can. And maybe you are there's so much we can do, because we have to be independent, but still we are there are several routes you can for interaction and for formal support.
Thank you. I think that was very interesting information for a lot of the people on the line that comes from the national academia environments, and also from people that have just started looking into how you can develop some concepts and ideas into a product for the future. So thank you so much for your presentation. Our last speaker today is Dr. Asbjørn Stray-Pedersen. Stray-Pedersen is a specialist in medical genetics and also the leader for the Norwegian Association for Medical Geneticists. She will present a clinician's perspective on the development that we have seen for people with rare genetic disorders with regards to gene therapy. So the floor is yours, Asbjørn.
Thank you. Can you all hear me?
We hear you very well, and we see your slides in not in presenter mode.
Okay, I'll check that out. Wait a minute. How about now? Is it okay now?
Now it's perfect.
Okay, so before I switch to Norwegian, I want to thank Martin Schultz and Clara Herbert for their wonderful talks and overviews and detailed information. The title of my talk was in Norwegian, so I decided to talk in Norwegian, and I can be quite quick, but I hope you can stay until the end and we can have a discussion where you can participate together with me if we will have a joint discussion. So then I could switch over to quick Norwegian. Takk til LMI og SLV for at jeg får lov å presentere dette her. Jeg en kliniker, men jeg representerer flere. Samtidig så dette et syn som jeg har, og som jeg tenker dere må se det ut. Dette en klinikers perspektiv. Det kommer ekstremt mange nye behandlinger nå, og de banebrytende. Det for tilstander, sjeldne tilstander som ikke har hatt noe behandling før. Det potensielt kurative behandlinger. Det klart at vi ønsker å ta dette i bruk. Og vi ønsker fra klinikkens side å bidra til oppfølging og registrering der det trengs, og det helt fantastisk å kunne være med på å følge barn og unge som blir gradvis sykere og mister funksjon, til nå å gi dem en potensielt kurativ behandling. Det noen utfordringer, og først og fremst må vi rigge oss bedre. Det har vi sagt en stund, men det gjelder enda mer nå, som det kommer som en stor bølge, disse nye behandlingene. Beslutningsforum godkjente medikamentet som Carla snakket om i sted, og det ble da verdens dyreste medisin. Tidligere så var det andre terapier genterapier som har vært kalt verdens dyreste medisin, og fortsatt det flere som kommer som enda dyrere enn denne Zolgensma, som ble godkjent da til bruk i Norge i oktober. Og det som helt fantastisk at Vi har hatt behandling for spinal muskelatrofi en liten stund, avanserte behandlingstyper, men dette her, denne one shot, en injeksjon, kan være potensielt kurativt. Det er helt fantastisk. Martin fortalte en del om virkningsmekanismen for AV, altså adeno-assosiert virus, vektorbasert terapi, og det er jo denne behandlingen. Men det som er spesielt, og hvorfor vi ser nå AV-virus, vektorbaserte terapier som kommer, er fordi at de er ikke replikerende. De ligger som et episom inni cellekjernen og produserer da transkriberes til protein uten at det i stort sett integreres da i genomet til cellen. Vi har også sett, når vi har tatt i bruk solgensma, som har nå vært benyttet på noen pasienter, at noen kan ha i bivirkning i form av immunrespons, som trombocytopeni og også økte transaminaser. Solgensma har vært og disse nye terapiene kommer i nyhetene. Fredsprinsvinneren fra nå i høst, han hadde satt i gang en donasjon i forhold til den samme behandlingen for en familie i Russland. SMA, det forklarte Carla ganske nøye, så jeg skal ikke si så veldig mye annet enn at det er tap av nevroner, og det er progressiv, og det var den vanligste årsaken til tidlig barnedød. Det er ulike alvorlighetsgrad. Tidligere snakket vi om SMA type 1, hvor det ble muskelsvikt de første 6 månedene, og ikke klarte å kunne sitte selvstendig. Og så er det SMA type 2, som starter noe senere, og hvor barna ikke kan gå uten hjelp. Dette er uten behandling. Det har kommet da nye behandlinger siste år. I februar 2018 ble Spinraza godkjent. Det er en antisens oligonukleotid-behandling som må gjentas. Og det er intratekale, altså man må stikke nåler inn i spinalkanalen for å gi barnet behandling, og det må gjentas. Nå i januar ble Evrysdi en peroralt ASO-behandling godkjent, som gjør at dette blir litt mer tilgjengelig for pasientgruppen. Men det jeg snakket om, og som Carla snakket om, er denne genterapien som kom i oktober. Og når vi finner disse mer målrettede behandlingene, når vi har de, så må vi komme til tidlig, og da for å forhindre tap av funksjon, og her i dette tilfellet tap av nevroner. Og vi snakker om tidlig diagnose, helst før man har utviklet symptomer. Gjerne en nyfødtscreening. Allerede da vi hadde Spinraza, så var det viktig å få nyfødtscreening på plass. Og søknaden ble satt i gang, men altså over 2 år etter at vi hadde Spinraza, så fikk vi screeningen på plass. 1 måned før genterapien for samme sykdom ble godkjent. Men vi var først i Europa i forhold til dette med å innføre screeningen. Og nyfødtscreening da baserer seg på en blodprøve som tas av babyene 48 til 72 timer etter fødsel, og det er en blodprøve fra helen. Vi bruker da dette filterkortet med den tørkede blodflekken til å gjøre analysene. Og vi gjør både biokjemiske analyser, men for SMA var dette den første ren genetiske analysen som var gjort som første test. Og langt de fleste med SMA har en delisjon av SMN1-genet, og det er det vi måler med kvantitativ PCR. Vi har en norsk variant som vi også tester for, en missens-variant som vi tester for hos de bærere av denne delisjonen, men det er en unntakssituasjon. Og så i tillegg på nyfødtscreening så gjør vi også SMN2-kvantitering, og det sier noe om alvorlighetsgraden for de SMA-typene, og hvor kvikt vi må sette i gang med behandling. Mye tekst her, men det viktigste er at per i dag så screener vi for 26 medfødte alvorlige tilstander i screeningen, deriblant SMA. Og formålet med screeningen er å avdekke og forhindre alvorlig sykdom og død. Vi kan skrine for flere tilstander, og vi bør skrine for flere tilstander når det kommer nå banebrytende genterapier som kurative, og hvor det viktig å komme til tidlig. Det klart vi må skrine for flere tilstander. Vi var først ute i Europa med SMA, og vi siden 1. september og nå til 8. april har vi funnet 4 med SMA, der 3 behandlet med genterapi. Vi var også først ute med SCID-screening i Europa, og vi har funnet, i nasjonale screeningen så har vi funnet 9 med alvorlig immunsvikt, eller SCID, som det heter på engelsk. En type SCID adenosin-deaminasemangel med alvorlig immunsvikt, og den kan vi også oppdage på nyfødtscreeningen. Vi fant et barn i forrige uke på screeningen, og det siste barnet vi hadde funnet med ADA-SCID i Norge, dette jo sjeldne tilstander, det var da rett før vi begynte med screeningen. Og det finnes 3 behandlingsmuligheter ved ADA-mangel-SCID. Det hematopoietisk stamcelletransplantasjon, det tar man ut blodstamceller fra donor, og etter at barnet har fått cellegift, så tilbakefører man det via en vanlig transfusjon. Det finnes enzymerstatningsterapi som vi kan holde det gående med en stund, men for de som ikke har donor, så ex vivo genterapi med lentivirus noe som har vært forsøkt og vellykket. Og det siste barnet før vi satte i gang med screening har fått genterapi. Og hvis dere vil se nøye på og få litt mer informasjon om det, så finnes det altså en NRK-reportasje fra mai 2018, og der det altså en jente med alvorlig immunsvikt som behandlet med genterapi. Og ved genterapi for ADA-SCID så henter man altså ut barnets hematopoietiske stamceller, og man behandler dette en ex vivo terapi, og så behandler man de med eller lentivirus som infiserer cellene og gjør de friske, og så tilbakefører man det til barnet med en vanlig blodoverføring. Det ex vivo, altså man arbeider med cellene utenfor kroppen, og så det in vivo, som da Zolgensma representerer. Det finnes også, det jo først og fremst disse her tingene som i bruk, som jeg nevner nå. Og jeg jo mest opptatt av de pasientgruppene vi følger. Jeg følger i vår praksis, og det jo sånn som ADA-SCID, det SCID-pasienter, det de som har eller SMA, men det finnes jo også mulighet til å gi spesifikk terapi rett inn i organet, og spesielt i øyet, som et lukket organ, så dette en metode som har vært prøvd ut med suksess. Og Luxturna var det første genterapien som ble godkjent i Norge. Og det skjedde i april i fjor. Og det var også medieoppslag på før den behandlingen ble godkjent, og pasientgrupper og andre ble veldig glade når dette ble godkjent. Det var helt fantastisk. Så avanserte terapier, da fremtiden blitt virkelighet. Vi har mange nye behandlinger for sjeldne sykdommer, og det det at de potensielt kurative med kun 1 behandling, og det helt fantastisk. Det handler om å finne de som trenger behandlingen raskt. Tidlig intervensjon gir bedre effekt og utkomme. Langtidsstadiene mangler fordi det en ny behandling, og det en risiko, og den risikoen når medikamentene eller behandlingen dyr, så veier den ofte veldig tungt i forhold til å innføre disse behandlingene. Men det handler om tilgang til behandling for våre pasienter, og ventetiden før de får tilgang uutholdelig. Også være på siden og være pasientens behandler, det helt forferdelig. Og vi vet også at jo mer tiden går, så det noen som ikke får. En ting som jeg også skal si litt om at dette sårbart siden det sjeldne tilstander. Og ofte det kun ett firma som utarbeider eller designer terapien. Og de også sårbare firmaer. De terapiene som, nå nevner jeg noen navn her, men det det for vi har i praksis i klinikken. De terapiene som aktuelle nå i pipeline helt tidlig, det en liten liste jeg laget her. Det kommer mange nå, det mange kliniske studier på gang, og det eksploderer. Så vi ser at det kommer til å komme en bølge. Og så spør jeg meg om vil det bli uetisk og ikke å ha screening for noen av disse tilstandene når vi kan forebygge alvorlig sykdom. Hvis vi ser på de tilstandene som søkt om eller tilgjengelig i Norge, så det rett og slett bare 2 som godkjent i beslutningsforum. Stimvelis har vi gitt til en pasient, men det den har ikke vært søkt om til nye metoder. Og så det dette lille firmaet, Bluebird, som har trukket seg fra Europa, fordi det har vært for vanskelig å få ting inn. Og dette ikke det første firmaet som har trukket genterapi fra europeisk eller fra markedet. Og dette kan vise et eksempel på at vi må også ta dette i bruk når det kommer nye behandlinger, for det ikke sikkert at det så veldig mange andre som kommer til å utarbeide genterapi for eksempel XALD. Vi har ordninger for hvordan nye medikamenter eller nye behandlinger skal komme inn i Norge, og på gruppenivå så det systemet nye metoder, hvor alle helseregionene, lederne i helseregionene sitter og bestemmer om dette noe vi skal innføre, men selvfølgelig etter faglig vurdering og legemiddelverkets vurderinger. For sjeldne små pasientgrupper det vanskelig med sikre data for hvordan det vil fungere, for vi har ikke de store gruppene, og vi har ikke heller langtidsdata, for dette jo nye behandlinger. Så utfordringene vi har fordrer nye strategier. Kanskje vi må drive mer med risikodeling, utfallsbaserte prisavtaler, sånn som det ble på Solgensma, og midlertidige innføringer. Mer samarbeid med industri for utvikling og tilgang til behandling og deling av data. Selvfølgelig skulle vi som sitter på klinikken, som ser at det samme potten vi nå har å rutte med når det kommer nye fantastiske behandlinger, og vi ikke har en økt pott som skal fordeles mellom pasientene i Norge. Og da må jo håp og forventninger justeres, også fra oss fagfolk. Vi ser at vi har muligheter, vi kan behandle, vi kan kurere, men dette må justeres hvis ikke vi kan få det til med innenfor det vi har fått muligheten til. Og så tenker jeg som så at nye kurative behandlinger og nyfødtscreening tett forbundet, og det må være mer synkronisering i forhold til det, fordi har vi en behandling så må vi finne de som trenger den, og I hvert fall hvis behandlingen godkjent. Takk.
Thank you, Asbjørg. We are coming to the end of the meeting, but I would like to ask you all if you could discuss a bit more just briefly, how could we collaborate better to enable patients with these diseases to get access to therapy? So, Martin, if you could say something about how you think the collaboration can be built stronger and how we can work together to have like a sustainable healthcare system that allows entry of these treatments so that patient can get access.
Yeah, thanks, Hege. And I think that might depend a little bit on the country where you're at, but I think meetings like you are having right now at this very moment are extremely important to bring people together so that people also start to know each other, that they know what maybe the concerns are or the challenges that the individual stakeholders have, and then that they're able to to work together to resolve this. I think what's very clear to me from all the other great presentations that I saw is that we are all in one boat. Everyone has the same goal. Everyone really wants to help these patients. So it's really just really this connecting and finding solutions together that needs to happen.
Thank you. Carla, can you say something about, because you are a part of the team approving a product for giving marketing permission, but then afterwards you have the reimbursement process. And I know there's been some discussion that the distance between the requirements in those two processes have maybe gotten too broad. Is there some discussion on EMA on how you could make it easier to go from one to the other, or how, because you are approving on the efficacy and the safety and the data for this one, but then we end up maybe with a bigger hurdle after the marketing approval now?
Yes, I think you have noted that. I think in the very well in the attempt to get products earlier to the patients, we have now systems like a conditional approval where we can make a benefit risk on data we find not fully comprehensive. We want some more data, but that can be provided post-marketing, and this is data which is very important for, let's say, cost effectiveness, or have a better clue on what the product really does. I think we should also understand that there's a difference between making a benefit risk assessment and making a cost effectiveness assessment. For a benefit risk, if you think about, for example, hemophilia, it could be very well, you could very well consider a benefit risk positive if a patient is 2 years off treatment for prophylaxis factor prophylaxis. However, if you want to pay for this, you might think 2 years is not sufficient. We want 5 years, or we want 10 years data. So there is a difference, of course, between a benefit risk and a cost effectiveness. And I think that this is something we should understand. But I think I agree that by giving making opportunities better to enter the market earlier with things like a conditional, this makes the problem for health technology assessment, the cost effectiveness assessment, more challenging, and this is particularly the case for these type of products, which is a one-time off product. So if it doesn't work, still you have to pay, and you have to pay for one treatment instead of, you know, if for oncology or for some other products, if it doesn't work, you stop, and you pay for each separate, let's say, dose, and here we have one dose, which makes it more complicated. And I think it's very good to talk to each other to understand what we expect, what type of data we expect, and also that there are differences, and that we are and we should be clear on we should find each other somewhere, but also acknowledge that we are looking at different things or expecting different things. And this is why I agree also with Martin that we should talk to each other. We should, for a benefit risk assessment, it can be very helpful to have patient involvement to really understand whether there is a benefit or how this benefit is experienced. And I think this could also help for health technology assessment as well. So quality of life is also important there.
I think that's very important. And I know that in the Swedish Medicines Agency, they made a report now on ATMPs last year, and they said during the discussion leading up to this, they said that maybe we don't add everything we need to into the health technology assessment, that maybe the patient perspective is not taken into account to a large enough degree. Martin, some more comments from you?
Yeah, no, I just wanted to make one comment, because Carla raised a very important point of uncertainty, right? If we want to bring these therapies to the patients early, the patients that direly need it, there will be some degree of uncertainty. And I just, I just wonder if real-world data is able to mitigate that, at least to some extent. And I think Carla very nicely talked to what they did at EMA with regard to that. But also, I think for payers, when you think about potential value-based agreements where you really connect the reimbursement to how well the patient is doing over time, I think that can really mitigate some of these uncertainties that are, of course, important on the payer side. But again, for this, I think we need to, rethink some of the policies that we have and make sure that we are able to, to have agreements like this.
Thank you. And I think that it's very important to start a dialogue early, both with the clinicians, with the payers, and between every stakeholder, so to speak, because if you're going to do this kind of follow-up, you also need to make sure that you have the possibility to follow up in a manner that brings you the answers you need, and that both the payer and the company can trust the data that are collected in a way that this is making a good agreement possible. So Asbjørg?
I was just wondering if there are European initiatives that look into this real-world data. Since we have the problem, each single country have the problem too, that we are not letting our patient getting access to the new treatment unless it has gone a few years. So how can are there good examples from some European countries that you have this real-world data integrated in the medical record, following up the patients to get to reduce the long-term safety and follow up? You can get the follow-up data and provide it to the industry, so we can have this conditional implementation of treatment earlier.
Martin, would you like?
So we can learn from each other. That's why I'm thinking.
Yeah, happy to comment on that. And Asbjørg, I think you make a really, of course, this is a really important point, and ideally, in particular, when it comes to rare diseases, we want to have maybe bigger registries than just single country registries that all have maybe all collect different data points, and it's really hard to make sense of the data. So I think there are some examples where things seem to come together very nicely. So for example, in hemophilia, there's the World Federation of Hemophilia, so the global hemophilia patient organization that works together with a number of pharmaceutical companies that all develop gene therapies, and they've had meetings with FDA and with EMA and with different other stakeholders. And here, the goal is really to pull together a global registries for all these patients, because again, just to your point, when it comes to durability of effect, but of course also safety events, what you want is a big pool of the biggest pool that you can get of data, similar data entries that are collected in different countries. So I think this is probably a very good example of a global registry for a rare disease.
Thank you. I think we are running out of time, and as usual, we have too little time for the nice discussion at the end. It's the way we always think we have a lot of time, and then we end up with really interesting discussions at the end. So I'm sorry for that. So I'll just briefly put up the slide for this meeting series and remind you that our next meeting will be on cell and tissue therapy, and it will be in June. The date is not formally set, but we will make sure to make the data available as soon as possible on lmi.no and also on slv.no and through social media. So we hope to see many of you on the line again then for the next meeting that will be in June. And I think this shows that we have still a lot of things to discuss, and that the value of this discussion should not be underestimated, because I think this is where the solution can be found. So we are working now together with Asbjørg and some of the companies already, starting to see if we can formalize a more extensive dialogue to see how we can help Norwegian patients in this aspect get easier access to these therapies in the future. So I think this is very important project that is ongoing now in Norway. So thank you to both Carla and Martin and Asbjørg for joining us and presenting their thoughts and knowledge on this topic today. And I wish you all a very happy Easter and a very nice weekend when the time comes to log off today. Bye.
Thanks for the invite again. Bye-bye.
Thank you. Bye-bye.